Good afternoon. Welcome to my Facebook group, Grow Your Own Science and Maths Like a Superhero. So this is my next video of the day. I'm going to be going through a bit of proteins and enzymes today. So first up, we're going to consider what proteins actually are. So proteins are macromolecules made up of amino acids, carbon, hydrogen, oxygen, and nitrogen, and some sulfur and phosphorus. So below we've got the typical amino acid structure. So if we're looking at the chemistry of amino acids, so we've got a typical carboxylic acid group here, and this is the amino group. So the typical example here, this one is glycine. Particularly important one because it acts on various different receptors. Which brings me to the next. So there's 20 amino acids. Okay, this is the chemistry of each individual one that occurs naturally in the human body. So these are the building blocks of all human life. So amino acids combine in various different molecules. So the example, the one that we just looked at, was glycine, which is one of the simpler ones. So this one here. So there's lots of others. So we've got alanine, tryptophan, phenylalanine, lysine, serine, threonine, cysteine, methionine, asparagine, histidine, arginine, glutamic acid, aspartic acid, and many more. So more recently, the one down here, asparagine, this one here. So that's come to attention recently because it's a mutation that occurs in COVID. And there's also a couple of others. Okay, so that's the chemistry. So this was a very good chart, comes from a website called compound.chem. Okay, so they will have slightly different properties. At the top is the key to what this chart shows. Aliphatic means straight chain, aromatic means like a benzene ring, it's a circular chain of hydrocarbons. Acidic, basic, self-explanatory, hydroxylic, and so on. So these are all basically what the chemical composition of amino acids are. So if we go briefly into glycine, which is the first one we mentioned. So let me just go back. So glycine, got your amino group here. And if we picture carbons here, so that is glycine, also called aminoethanoic acid. So on the first recording today, I touched on how hydrocarbons are named. So this particular one, glycine, was discovered by a chemist in 1820 in France, and he hydrolyzed gelatin by boiling it with sulfuric acid. So basically he reacted gelatin with sulfuric acid. Okay, so let's move on. There's a brief mention here about where the name glycine comes from. It comes from a Greek word. Okay, so let's go into a little bit more detail about amino acids in animals and plants. Plants synthesize all the necessary amino acids they need, but animals do not. Amino acids in animals can be divided into these different categories. So essential and non-essentials. So essential being ones that we can't make, but we get through our diet, or non-essential ones that we can make in our bodies. Quick mention about the bottom types. So there's 10 different types that was outlined on the previous slide. When we're talking about what sort of chemical properties amino acids have, we refer to it as some of them that can behave as amphoteric. Now, what does that actually mean? So amphoteric is basically means amino acid can act as either an acid or a base, meaning it's got a transferable hydrogen proton and an atom with a lone pair. So the examples here are hydrochloric acid and water rather. So amino acids in nature many of them occur, and they combine to form things like dipeptides, polypeptides, lots and lots of different molecules. So we've got the example it gives here is what happens when two glycines react. And if you see here, this is what's called a condensation or hydrolysis reaction, depending on which way it goes, taking water out or adding it in. And you can see clearly here what happens when it joins to form a dipeptide. Okay, so basically water is taken out in this particular reaction. So you've got a glycine plus glycine. So you've got two glycine molecules. And you can see here where the water is taken out to form the dipeptide of glycine. Dipeptide just means two proteins joined together. So further reactions will just cause amino acids join in what's called a polypeptide chain. So a polypeptide is basically just a long chain of amino acids. So we covered briefly in my first video about DNA and DNA synthesis of proteins. So this is an important concept to understand. On to our next slide. So let's have a quick look at protein shapes. This is the key to how we understand receptors and enzymes in pretty much every protein there is in the human body. So we're going to have a look at levels and structural organizations of several different proteins. So the primary structure refers to the number in the amino acid sequence of a polypeptide chain. One peptide bond is present. For example, the example here is lysozyme enzyme, which is in tears and saliva. And you can see where the dipeptide bonds occur in the polypeptide chain. So the next one we're going to consider is the secondary structure of proteins, which is helical shapes or sheets. Examples of these are collagen and keratin. And if we think about what these actually do, so collagen naturally occurs in skin. Keratin is actually spelt wrong here. It should be with an IN on the end. And this is what they actually look like as chemicals. So you've got all of these carboxyl groups that have formed dipeptide bonds and layer upon layer of sheets. So the next one we're going to have a quick look at is the tertiary structure proteins, which are basically referred to how proteins bend and form different shapes. So the following types of bonding are present here, which are peptide bonds, hydrogen bonds, ionic bonds, and disulfide bonds. So if we think back to different bonding types, disulfide just means basically bonding between two sulfur molecules. Peptide bond I explained before. Hydrogen bond obviously occurs with hydrogen. And you've got all of the different relationships and bonding types in proteins which contribute to basically how they are formed and their stability. The quaternary structure is the final sort of class of protein structure, which occurs in mostly globular proteins and molecules, like say myoglobin, which are bound largely by hydrogen bonds. So you can see here, a good example is hemoglobin, which has lots of different subunits, and obviously it basically carries oxygen around the body. The structure of these proteins is basically what gives them their function. So how do we identify them? So the example here is a biuret test. So a biuret test is just mixing potassium hydroxide and a little bit of copper sulfate, and you basically get a little bit of coloration of purple to detect a protein. So let's go a little bit into the functions. So the examples we gave here were keratin and collagen. Pepsin are also amylase, which is present in saliva. So the first class is structural proteins. So like we said before, we've got several of these will occur in plants as well. So you've got collagen in the skin, keratin as well, and quite often you'd find these are present in different cells like keratinocytes. And we've got all the other different enzymes as well. So amylase, lipase, pepsin, trypsin, and chymotrypsin. So basically these are mainly digestive enzymes. So let's go a little bit into the other types. So we've got transport proteins, protected proteins, and contractile proteins. So obviously a key point at the moment is function of different proteins in the human body. So I'm going to talk a little bit more in one of my later talks about antibodies, which is my field of interest. But the other different protein types, they all have to do different things. We've got hemoglobin, hemocyanin, albumin, and the protective proteins. So these are involved in clotting, fibrinogen and thrombin. And the contractile ones are more involved in muscles. We have actin and myosin here. So you can get a good idea of what the protein shapes are. And it's important to bear in mind those protein shapes are what give the proteins their particular functions. So we've got storage proteins and toxins next. So before we should really move on to enzymes, the key to understanding metabolic reactions. So broadly speaking, enzymes are proteins which belong to organic compounds naturally made by us, so people. And living cells can contain up to hundreds and hundreds of different types of enzymes. And they're exactly the same, or they are globular proteins. And if we go a little bit into about what they actually do, they generally work at between 5 and 40 degrees. So they increase the rate of reaction without actually being used or take part in it, and they don't have any changes during the course of the reaction. It's important to bear in mind that enzyme activity is dependent on temperature, pH, and lots of different things. And we refer to very high temperature denaturing enzymes because it basically changes the shape of their protein, so they can't actually do basically what their job is in the cell. So as an example, enzymes are specific and generally catalyze just one particular type of reaction. I'm going to mention in my later talk about what ATPase is as an example. And basically it's a bit like a lock and a key. So an enzyme has what's called an active site, and all an active site is that particular protein shape, and the substrate it acts on basically slots in and causes a chemical reaction. So originally, Emily Fisher in 1890 proposed her lock and key hypothesis, and that was a very good one because it basically describes exactly what happens with enzymes. So a substrate attaches to an active site, which activates itself and forms products like this. So you get your enzyme substrate complex and the two products being released of that particular reaction. So the next theory was Koshland's induced fit. I'm not going to go too much into these, and basically that's a quick detail. So factors that affect enzyme activity: cofactors, inhibitors, pH, temperature, and obviously the concentration of substrate or the enzymes. So this is examples of what happens. So generally enzymes will have an optimum temperature. Examples of that are numerous. If it was in the intestines or the stomach, they would have an optimum pH of somewhere down here, 3 or 4. Cofactors. So these many enzymes need non-protein helpers to their catalytic activity. They're divided into 3 groups: inorganic ions, coenzymes, and prosthetic groups. And I'll come to examples of those later. So on to some more information about what affects enzyme action. pH I mentioned. So pepsin is a typical enzyme in the digestive system, and it has an optimum pH of here 2. And the reason it is because the stomach is rather acidic, so it wouldn't be able to work if it didn't have an optimum pH around there. Whereas amylase in the salivary glands has an optimum pH of around 7. Temperature, well, the human body is naturally 36, 37 degrees. Go outside that range and enzymes don't work as well. And it basically refers to the shape change causing loss of function. This is a good summary about the proteins. So only in 5 or 10 minutes, but we've gone through primary structure, secondary structure, tertiary and quaternary structure, and some different facts. Now I'm going to go into a lot more detail about antibodies and a few of the other things that proteins are important in, but this is probably the most important part of research in molecular biology, because proteins make it nearly everything they do make everything in the human body. Anyway, this was my 5th recording. I hope you enjoyed that, and see you all soon.